Part of what we do is create these moths or these flies which have new pieces of DNA in them.
Yeah.
Because we want to sort of follow and study proteins that are in the cell.
Yeah.
So to do that, you need some way of highlighting those proteins, and we usually do that with a fluorescent tag. So that's why we end up with flies that have particular proteins in them that glow.
Yeah.
So in order to do that, we need to get these pieces of DNA stably into their DNA.
Yeah.
So we take the piece of DNA that we want and we inject it into very early embryos before their cells have properly formed, and some of that DNA gets incorporated into cells.
So you literally just inject it not into a particular place? Do you have to get it the right place, or does it find its way there?
No, it's better if you inject it into the area of the embryo where their pole cells, germ cells, what would we want to call them? The cells that are going to become the gonads, so the sperm or the egg, because we want that incorporated into those cells because they're the only cells which are going to get passed down through the next generation.
Yeah.
So
this has got nothing. You're not using CRISPR technology. You are. Oh right. Okay.
So, we're using CRISPR to do that. We use what we call standard transgenic tools for insects, which is a system called transposon-mediated transgenesis.
I've actually heard of that.
Okay. So we use that quite a lot. That's our standard technique.
Yeah.
But more and more these days we're using CRISPR, which replaces pieces of their normal DNA with these additional tagged versions, or we can knock things out, or we can add things. So this microscope here, you can see this little apparatus here, that terminates with, I won't put one of those out from there. So in here we have little injection needles. So within there's a very fine needle. Okay.
Oh, I see. Within
that kind of plastic tip.
What sort of width? I mean, how fine? Do you know? I mean, I'm not going to be putting that in the song, but I'm just being curious.
Yeah, tiny. I can find out, but I don't know what they are. So we put our little bit of DNA in there, and we attach one of those needles to the end of this. So it sticks out about there, and then we have a microscope slide which will have our embryos on it, and then we will go through and we will inject between 20 and 40 embryos per slide on there and put the DNA in them. This is an automated thing called a femtojet, so that will deliver 5 nanoliters of DNA into every embryo, so it's kind of part manual and part automated.
That's a billionth. Is it?
Nano 10 to the minus 9. So yes, billions.
Yeah, billions.
Yeah.
Okay.
In there. So that's how we get the DNA in, and then we have to wait until those flies or those moths become the next generation, and then usually if we're screening them for a fluorescence, so we've added this little fluorescent tag to them, then we'll use this microscope here which has fluorescence on it, and we'll put the flies or we'll put the larvae down on a plate on there, we'll shine fluorescence at them, and hopefully if they've incorporated the piece of DNA in them, they'll glow.
Then where does that fluorescent DNA come from originally? Or is it...
So the fluorescent part, so actually look, you can already see it here. So the piece of DNA, so if we think about the chromosome, this is a linear chromosome, this pair of chromosomes, right? Think about that. So say that what we've done is we've inserted a piece of DNA in this location.
Mm-hmm.
So if we were to zoom in on what that piece of DNA looks like, so it's it's a double helix, right? So it's the double helix DNA. Okay. It has the copy of the gene or the protein So it's the gene that encodes the protein of interest, whatever that might be. I don't know, what should we call that? Or I'll call it tubulin, okay, which is the protein which is going to make these elements in the cell called microtubules, which are important for cell division. And then upstream of that, I've done the wrong colours, but hey ho, we will have the gene for a fluorescent protein.
But what I meant was,
So this,
where does that gene originally come from?
originally came from jellyfish.
It
That's
did,
the one from
okay.
jellyfish.
Yeah.
So
Yeah,
that's
okay.
so that's called green. Well, I know I've done it the wrong colours. That's called green fluorescent protein, and that came from a jellyfish called Cora victoriana, I think. Yeah, so it comes from a jellyfish, and that's the gene that encodes that. And there are various other ones.
Now, can I just check? Is that actually a jellyfish gene in itself, or is it something else that lives in the jellyfish?
No, that's an actual jellyfish gene.
Because some jellyfish incorporate
Yes.
stuff, don't they, into their bodies
yes, there
that
are,
glow.
and there are other ones. So
Yeah.
there is, so there's a red fluorescent protein, and that comes from discoidum something or other. I can't remember.
I mean, it doesn't really matter.
But yes,
But
yeah, so
all
there's-
these
in-
terms
in
can be
nature,
useful in a
yeah,
song. It's
in
kind of,
nature, there are
yeah.
a variety of fluorescent and iridescent, and what's the other one?
oh, I've written 2 songs about this. So.
Yeah, proteins, genes. So yes, we take that one.
Yeah.
Because of its.
Not that I have a lot of detail in my songs, we know, but yeah. Oh, fascinating.
Okay.